Hey everyone, this is the introduction to Lab 22 on red blood cells and blood typing. This follows labs in your FOX 6.1 with red blood cells and oxygen transport. So you will be learning how to do hematocrit as well as doing counts for red blood cells, as well as finding out hemoglobin concentration, and then you will be able to calculate these values with the data that you obtain from the lab. Also, FOX 6.3 is all about blood typing, finding out what determines RH factor, as well as how to do ABO blood typing. So of course, if we were doing this in lab, we would have to always follow our universal precautions and just assume that all material is hazardous. Always wear gloves, especially when working with other students' blood. And we always want to properly dispose of clean, contaminated materials. We know that lancets and contaminated sharp or hard objects go into the sharps containers. Soft, contaminated materials such as alcohol wipes, gauze, band-aids, and paper towels will go in the biohazard bag, and no other trash will go in that biohazard bag. And then reusable contaminated equipment we can wash with amphil, such as test tubes, beakers, pipettes, and hemocytometers. We don't want to wash blood contaminated equipment for anyone else, so if you yourself are donating blood or using your blood for the experiment, only you will wash that equipment. And of course, if we were in lab, there would be no eating or drinking when we are working with blood. So the first part of this lab, which is from FOX 6.1a, is doing a total red blood cell count, or RBC count. So we actually performed taking blood and filling a capillary tube earlier this semester. So first we would obtain whole blood, filling a small capillary tube. So we would have the volunteer sterilize their finger or use an alcohol swab to clean the finger. We take a lancet, poking the finger, and then filling up a capillary tube. We want to make sure this capillary tube is filled completely, and it's always good to kind of hold the capillary tube at an angle to allow for filling of this tube. We would then take this tube and shake it into a centrifuge tube. So this small centrifuge tube here, we would use the capillary tube and shake it into this centrifuge tube. And then once it is filled, we want to shake the centrifuge tube for about for a little bit, and then let it sit for 1 minute. Now, there's a special lysis solution within the centrifuge tube which will remove other cells other than red blood cells, because we want to isolate those RBCs. We will then, after sitting for 1 minute, we will use a pipette, a micropipette, to add this blood to the hemocytometer. We'll put a cover slip on top of the hemocytometer. It won't cover the entire surface, but you want to make sure that this cover slip is centered. We'll then add the blood with a micropipette to both V slots, allowing for that capillary action to move the blood onto the grid, at which point we will do counting of the red blood cells. So here we see our hemocytometer. You can see that there's two V lines at the edges of the hemocytometer. The cover slip goes on top, and then we will pipette the blood into these introduction points at the V points. The cover slip on top will allow for that capillary action to disperse the blood. There is a mirror located on the hemocytometer that contains a counting grid. So the counting grid will look like this. What we want to do is we want to count 5 small squares. So first we have a 5 by 5 grid, and within that 5 by 5 we then have a smaller 4 by 4 grid. So we want to count the cells in squares 1, 2, 3, 4, and 5. So within each numbered square, we can see that, again, there is a 4x4 grid. So it's going to take a while. There are a lot of cells, and we want to count the cells that are, of course, within the 4x4 grid, and then you can look at the 4x4 grid and determine what cells you want to count that are on the lines. We're going to assume that there is an equal number of cells that touch the lines, and we want to take the average of that. So we're just going to count the cells that touch 2 lines. So you can pick any 2 lines, either, you know, the ones on the sides or ones that are on the side and on the top or side at the bottom. It's up to you, but just you pick 2 lines, and then you say, okay, any cells that are touching these 2 lines you're going to count. You're going to include in the count, and the other 2 borders you're not going to count. So you're going to count the cells within the 4x4 grid. It's going to be a lot. There expect to be about 80 to 100 cells per 4x4 grid. And you're going to do this 5 times. So you're going to count all the cells in box 1, 2, 3, 4, and 5. So once you have gotten this number, you are going to add up all the cells that you counted in square 1, 2, 3, 4, and 5. You're then going to multiply by 10,000. Now 10,000 is the dilution factor. So for example, we're just going to use a round number. If there are miraculously 100 cells in each square, so 100, 2, 3, 4, 500. If there are 500 cells that we counted in all 5 squares, we'll multiply that by 10,000. So 500 times 10,000 will equal the total number of red blood cells, and that is equal to 5 million, because 500 times 10,000 is about 5 million cells per microliter. Now it's important to note that, of course, this number is going to be a little bit bigger for male patients or male volunteers, and female patients or female volunteers, they're going to have a lower number of red blood cells. So that is the experiment on counting total red blood cells. The next part of this lab will be taking a hematocrit. So again, we're going to fill capillary tubes, larger capillary tubes, and these are heparinized and then you want to make sure you fill these capillary tubes at least half full, but more, of course, is better. And then you will want to seal one of the ends with a cryo-seal clay. So we place it in this little container here, and we twist it, and it will seal off one of the ends. We then centrifuge, so we place these tubes in the centrifuge. I remember a lot of people had problems with this because it wasn't placed properly, or it wasn't balanced, or the centrifuge tubes exploded in, or the capillary tubes exploded in the centrifuge. But luckily you don't have to do it, but know that once we centrifuge these capillary tubes, we can see a separation of the whole blood into its component parts, into the plasma top part, and then there will be a buffy coat, and then the bottom part of that capillary tube, hopefully, if it works out, will be packed red blood cells. So we then take this capillary tube and we place it on a hematocrit wheel. So we want to make sure the top part where the plasma starts is at the number 100, and then we want to place where the top of the red blood cells that you can see in the capillary tube is placed at this line, and then it will give you the percentage of red blood cells within that capillary tube. So we talked about what the normal values were for hematocrit. Normal is about 42%. In men, it could be higher. Women, of course, it could be lower, but the magic number we're looking for is about 42%. When we talked about anemia in our lecture, we said that less than 37% could be indicative, or you could use that to help diagnose anemia. Okay, so that is how we take a hematocrit value, and of course, this is in a it's in a percentage. The next part of the lab is getting hemoglobin concentration, and this is the Tallquist method. Basically, you add a drop of blood. Once you lancet a finger of a volunteer, you add a drop of blood to a piece of bond paper, and then you let this paper dry. Once the blood has dried on this paper, you then go outside or go where there is natural light, and you compare the color of the drop of blood to this reference scale. So you can see that the darker the drop of blood, the higher the hemoglobin concentration versus, the lighter end, which would indicate a lower hemoglobin concentration. So you want to make sure you are able to compare the color of the drop of blood on the test paper with the color of the reference scale, and you can make your best guesstimates. So that is how we would be measuring hemoglobin concentration in the lab. And the normal range that we're looking for in males, typically hemoglobin is between 14 to 17, and with female patients, normal range of hemoglobin is 12 to 16 according to your book. Of course, different labs and different books have different numbers, but typically hemoglobin below 12 is clinically considered anemia. So next part of the lab is blood typing. This is actually one of my favorite labs. With blood typing, we would get those blood typing cards, and with a lancet, you would take your volunteer's drop of blood, place a drop of blood in each circle. There are 4 circles, one with that's labeled anti-A, one that's labeled anti-B, one that's labeled anti-D, and one is a control. And of course, you'd label the blood typing card with patient's name as well as birth date. So you add a drop of blood to each circle. You then add antibodies about the same size as a drop of blood. The anti-A would be in a blue container, the anti-B is in a yellow container, and then the anti-D or the anti-RH, RH standing for the rhesus factor, would be in a clear bottle. So after adding the anti-A to the circle indicated, anti-B to the circle indicated, anti-D to the circle indicated, you then take 3 little plastic sticks that are, for each circle, and you would mix the antibodies and the drop of blood. You want to observe any agglutination. Agglutination is just a fancy word for clumping. If you see agglutination, that determines your blood type, and we'll talk about how we determine blood type with regards to presence of antigens, presence of antibodies in the discussion. So if you see agglutination, that determines your blood type. Notice that there is no clumping in either anti-A or anti-B. That indicates that there are no A or B antigens on the surface of the red blood cell. It's the antigens that determine the blood type. So if there are no antigens, this person would be blood type O. Now, anti-D, this determines whether you are positive or negative with regards to the presence of that RH factor or that D antigen. You notice that there is agglutination or there is clumping in the anti-D. So this person, along with being the O blood type, they would also be O positive. So again, the RH factor, if it is positive, it will react with the anti-D or anti-RH antibody. A person is RH negative if there is no reaction or no agglutination. So again, based on this card right here, there is no reaction in anti-A or anti-B, so the person is O, but they are positive for that anti-D. So this person would be O positive. So of course, with disposal, everyone in the lab will handle their own blood. Gloves should be worn, and you want to wear gloves for shared equipment and assume that it is contaminated. All lancets and slides will go in the sharps container. Any soft stuff like band-aids or gauze or plastic goes in the biohazard container. If there's any spillage or drops of blood on the bench, you of course want to clean with Anthil or Lysol. The Hemocytometer, as well as cover slips, will be cleaned with Anthil and bleach first, and then rinsed well, and then dried. You don't want to cover or throw away any of the hemocytometers or the cover slips because they will be reused. So I will be posting the blood typing worksheets along with this video. The majority of the video is about blood typing, what genotypes determine the blood typing, doing your Punnett squares, and that will be discussed in the discussion of this lab.